speaker is Isis, a PhD student from the University of Tennessee, and she's going to give us a talk on the biosensors for detecting nuclear production activity in the environment.
Hello, can you hear me?
We sure can.
Okay, and is my screen sharing?
Yes, you may want to go into the full presentation view, however. Perfect.
Okay. So yes, the title of my talk is Biosensors for Environmental Detection of Nuclear Proliferation Activities. And my advisor is Dr. Terry Hazen, and some of my co-authors are Martine Duff, Henrietta Dulai, Eric Alm, and Adam Arkin. Okay, is it not progressing?
Does not appear to be progressing on my screen.
Me either.
Yeah, maybe escape out and try loading it up again. Or is the screen frozen? There it goes.
Working now. Sorry about that. So the problem that this work seeks to address is in part that detecting and monitoring nuclear activities at undeclared and legacy sites is a particular challenge for non-proliferation technologies. So one of the solutions that we're looking at is that the biogeochemistry of microbiomes in plants happen to record contamination from both past and present nuclear fuel cycle activities. And this has been shown in a number of previous studies. So what we hope to do is sort of develop some of these biogeochemical models and proxies to help resolve the timing and extent of proliferation and other fuel cycle activities for both undeclared sites and legacy sites that have been used for various processes in the past. So in terms of the relevance of this work, we hope that the outcomes will help to establish environmental monitoring and biosensor technologies for helping to detect undeclared nuclear materials and activities, with the overall goal of helping to deter arms proliferation while also helping to support safe development of nuclear energy resources in the United States and abroad. So the technical approach that we are using, sort of broadly speaking, is to do biogeochemical analyses at sites with very long, varied histories of contamination and use for different fuel cycle activities. So some of the sites that we're looking at least initially, are Y12 and Oak Ridge National Lab in Tennessee, as well as Savannah River, the Savannah River site at the Savannah River National Lab. And then we hope to be able to expand to some other sites, including Los Alamos and Hanford. So our approach that we're going to start with is sample collection. So that's going to include plant, groundwater, and sediment samples collected from known and suspected contaminant pathways, and then use those samples to do microbial community analysis using 16S rRNA gene surveys, and then supplemented with higher resolution metagenomic and metatranscriptomic data to get a little more information about microbial community function and gene expression. And then as we move a little further along with the data, we hope to actually start looking at proliferation signatures within some of the samples. So we're going to do that by statistical analysis of the microbial communities, the plant biochemistry, and also sort of contaminant evolution using random forest and Monte Carlo models. And then as we move along, we hope to start teasing out some of the time signatures in the data and develop a temporal framework for some of these biosensors. And we plan to do this by integrating the historical records that we have available for contamination events, different bioremediation efforts at all of these different sites, and use that historical data to calibrate and inform some of our statistical analyses and models. So for the initial results, these are very preliminary. We haven't been able to get very far in our results yet, but we have identified correlations between 16S genes and at least 26 key geochemical parameters. And so those are shown over here on the right in the uppermost graph. So some of those parameters include uranium, nitrate, pH, and then some key taxa have also been identified that could, or at least have potential to be indicator species. So this includes Deinococcus radiodurans and Kinetococcus radiotolerans. And so those have been found in association with very high level radioactive waste. And then Microbacterium genre have been found in association with plutonium. And then we have other species like Geobacter, which have been found in association with uranium, iron, and Rhodanobacter in association with nitrate, which is often a co-contaminant with uranium associated with uranium processing facilities. And then for some of the potential plant biosensors that have been identified at Savannah River site, those are shown there on the bottom right. In those two photographs, they include American burr reed, and bladderwort. So we plan to sort of target some of those whenever we do our microbiome analyses from Savannah River. So the expected impact of this work is the development of biosensor technology for, as I mentioned earlier, detecting and deterring undeclared nuclear activities. And this would be based on sort of three different aspects of biosensor technology that we're looking at in the environment. So that'll include sort of broader biosensors based on microbial community function and dynamics, as well as more specific or biosensors that target more specific indicator genes or microbes. And then also the specific plant species and those biosensors. So sort of combining these three different technologies into a more comprehensive monitoring and verification technology to be applied to the environment. And these techniques, we think that they're going to be very simple and cost effective and very quick compared to some of the other technologies. And so in terms of the impact of the MTV project on my development, obviously it's helping to develop technical acumen and access to this whole network of experts in nuclear nonproliferation and arms control research. And then perhaps maybe one of the most important things is that the resources that are being shared by national labs like Savannah River and Oak Ridge are going to, or they're ending up being sort of core components of my graduate research. So this includes access to field sites, samples, analytical techniques. All of that's been very important. And then also fostering this collaboration and technology transfer with the different labs, Oak Ridge, Y12, University of Hawaii, MIT, and UC Berkeley. And so some of the next steps for the research are to actually get into the field and start getting some of the samples and looking at the data. So we hope to support some of the field work going on at the Savannah River site in terms of their sample collection and analysis efforts. And then we sort of want to build off of the experience at Savannah River to develop a sampling and analysis plan for both Y12 and Oak Ridge. And then we hope to sort of consider expanding evaluation to additional sites, like I mentioned earlier, potentially Los Alamos, Hanford. And I believe that is it. Does anyone have any questions?
All right, thank you for the great talk. So if anyone has any questions, they can go type into the chat or just speak up. Okay, so Sarah Potsi, she has a question. Hi, Isis, nice presentation. Can you say a bit more about the modeling you mentioned, Monte Carlo specifically?
Yeah, so I think whenever the hope is to use the sort of stochastic analysis to pull out some of the correlations between microbial community function, structure, and what have you, and some of the time signatures that we're hoping to tease out of the data, especially with some of the larger data sets that we might be getting as we combine data from different sites.
Are there other questions for Isis? Yeah, so I guess if not, I'll ask one kind of related to the previous. So have you looked at any algorithms for data mining from, let's say, machine learning and things of that nature to try to analyze these large data sets, or is that still something being considered off in the future?
So I personally haven't looked at that, but I know previous studies have, and I'm not sure if Dr. Hazen is on the call. He might be able to speak to that a little bit more, but I know we do plan to do that. That is sort of in our work plan as we start to get some of this data and work with it. Yeah.
All right, thank you so much. So we guess we do have a question. So decades ago, I remember reading that humic acids were important in uranium transport in the environment. Is this important for your work?
Yes, I think that certainly is something that we're going to have to look at and constrain, especially if, for example, that has resulted in transport out of the sort of contaminant pathways that we plan on sort of focusing on. But yeah, so humic or organic acids, as well as things like nitrate, they can all impact uranium mobilization. And so the concentrations of uranium that we end up looking at, we're going to have to sort of consider the effect of those types of parameters on the values that we're looking at in present day.
Thank you so much. Are there any other questions for Isis?